Now my beans regret nothing. Nothing.
Mr. Enchiladas, don't be too dramatic. This treatment, we can use antitoxin, which can help and neutralize Travis from binding to more proteins, and respiratory support can help with the treatment also. Let me take a look at you. No external wounds, no external wounds. Have you eaten anything weird as of recently?
Anything weird?
Hmm?
Oh, my judías verdes, how do you call it? Like my green beans. My tia made some green beans for my enchiladas. It's not kind of weird though. Yeah.
And class, that's how preservation of foods such as green beans is important, as it's a matter of life and paralysis. Meet our patient, Botulism in action.
Oh yeah! When I get the signal, I jump into action. Flexing is life. Oh no! Without net signal, no more flexing. Ugh, feeling weak. I can't contract, so breathing is a struggle. That's why patients sometimes need a ventilator.
She's a protein too. For example, if you look at a eukaryotic cell or bacteria in general, proteins are made, or specifically she is made in the ribosome, which are free-flowing or free-floating in the cytoplasm. But if you look at a human cell, which is comparable to us, it goes through a more scenic route. For example, proteins like the acetylcholine receptors, which go through the ribosome, to the rough ER, to the Golgi apparatus, to be packaged and to be made as vesicles.
Greetings, humans. I'm Travis Toxin, and I'm the main star of Botulism Botulism. My specialty? I stop nerve cells from communicating to muscle cells.
Wait, but I'm supposed to send acetylcholine in little vessels to the muscle cells so he can contract.
But wait, how did Travis get here?
And I need my cytoskeleton microtubules to transport these vesicles to the membrane. Whoa, slow down there, buddy. I cut up those snare proteins like butter. No fusion, no neurotransmitter release.
Is this what I think it is? And understanding that's why cell biology saves lives. Stay safe, study yourselves, and don't mess with home canned beans.
Thank you, Dr. Smart. Bye.
Bye.
Okay, student, I'll be asking you a set of discussion questions, okay? Ready? Number 1. Which type of proteins normally allow nerve cells to send signals to muscles?
Channel receptor proteins.
Good job. Alright, how does botulinum toxin interfere with this process?
It blocks release of neurotransmitter from nerve endings.
Nice. Alright. Outline the path of its synthesis in bacterial cells.
Made by ribosomes in cytoplasm, folded and released.
Perfect. Now, compare this to how a human would synthesize its own proteins for signaling.
Ribosomes on rough ER, Golgi for processing vesicles and secretion.
Perfect. Which human organelles are critical in protein processing and secretion?
Rough ER and Golgi apparatus.
Awesome. Alright. Which intracellular elements guide vesicles to the plasma membrane?
Cytoskeleton, the microtubules, and motor proteins.
Good job. How might disruption of these processes worsen the effects of botulinum toxin?
Vesicles can't reach membrane. No neurotransmitter release, and the stronger paralysis.
Right. Explain how the loss of signal release affects skeletal muscle function.
Muscles can't contract.
Exactly, that's why you can't move. Why does this lead to paralysis?
No nerve signal, no movement.
Right. How does the body attempt to compensate for this?
Increases nerve firing, recruits other pathways, but not enough.
Exactly. Why is mechanical ventilation sometimes necessary?
Breathing muscles can't contract.
Alright. How can understanding cell biology guide treatment strategies in biomedical science in general?
No acetylcholine release, then diaphragm and chest muscles fail.
And how does this relate to general cell biology treatment and like using strategies specifically in biomedical science?
It shows where the toxin acts and guides us on of antitoxins and support therapies.
All right, good job.